Pharmaceutical Processing, in conjunction with INTERPHEX 2016, presents INTERPHEX Live.
Okay, so next topic. We're going to be speaking about large-scale continuous processing in the age of single use, all right? We have Lynn Freck, Director of Sales from Powell Life Sciences. We got Mark Bishop, Principal Scientist, Powell Life Science. I can start by telling you, of all the topics we had, somebody robbed it, but anyway, we used to have this billboard outside, had all the top. I got more questions about that topic. It seems that people felt it was like a combination of incongruent terms. Large scale, continuous, and single use. So maybe, can we start there, please, so we can put these people at rest? I'm sure they're chomping at the bit here.
Well, I too was a little surprised by the title because
oh, okay.
It was not my title, but part of it is that things don't need to get very large in continuous manufacturing, although the system that we are showing in the Paul booth today is a system built to enable in a 2,000 liter plant of single use design to be able to capture in a primary chromatography step everything in 6 to 8 hours. So that's pretty large scale. If you go look at the skid with a single use flow path, you'll see what I mean. But to back up a little, I think you could think of continuous manufacturing as anything to kind of apply engineering efficiency as a means to maintain or improve product quality. That's kind of our broad definition. So we look at this as process intensification and how do you become more compact. And once you become compact, single use becomes easier. So you get kind of this dual approach where one thing feeds on the other. When equipment gets smaller, it's easier to make the product contact elements single use. So we've decided to employ a strategy in which much of the product flow path or all of the product flow path is single use. If the equipment gets smaller, that's easier to do. So that's kind of how the two things go together. Without continuous processing, it's difficult to make all steps single use.
Okay, so the word that doesn't really necessarily fit is large, right? I thought I missed the entire thing there. Okay.
I think when people were speaking about large, they think of what does it take to replace a 2-meter column in the largest scale biotherapeutic manufacturing plant that we look at. And it turns out in continuous chromatography, that's still a fairly compact system.
I got you. All right,
Yep.
You know what? Why don't we talk about the continuous and why single use makes sense? Why don't we start right there?
Yeah, so again, our strategy was to make all the product contact elements single use, and really it allows you to design configurable systems and facilities worldwide. One of the things we've heard from customers, they want to have in-country manufacturing, which Parrish was just mentioning, places where we can deploy full processing lines once the process is set up to be able to do that in many places, and single use makes that easier. So that's a big part of it.
So continuous, process, small, they all
kind of play off each other.
So as long as you take the word large out, you're good. Okay.
Yes.
So they are enablers.
Yeah.
Okay. So Mark, what's primary? Let's face it. I mean, as scientists, we're always looking to be in stasis. So what's the driver in your bioprocess to go to continuous?
Well, if you look at our industry is one of the largest industries who's in manufacturing, who's actually considering continuous manufacturing. Many industries have preceded us, including car manufacturing, food, chemical processing, and all of these industries did it for financial reasons. So their incentive was driven by making more products in a smaller facility, reducing the amount of auxiliary materials they use, and reducing waste, which in those industries is a big deal. For our industry, I would say manufacturing more products in a smaller facility reduces your capital investment. In our industry, our facilities are very capital intense. So to have the ability to produce more products per dollar invested is a compelling financial argument. It's not only that, if you combine it with single-use technologies, then the decision to invest can be made a little bit later at a point where the risk of the investment is, I would say, easier, manageable, could be lower. So the point at which you need to invest, and the investment is going to be lower, That reduces the risk of the investment, and that overall adds to the net present value of your overall program. While people were moving towards continuous manufacturing in the other industries, they also realized it was much easier to control their processes. Like in chemical processing, if you have to start up your plant and keep and maintain your operation at a certain steady state operation, that is so much easier than doing all these transient operations that we are used to in our biopharmaceutical industries. So it was actually an eye-opener for me as a chemical engineer to go into biotech and see all these batch processes. And I'm used to designing continuous processes. I'm used to designing steady-state processes. To get all of a sudden time entering into the equation make my life quite a bit more complex. So to me as a chemical engineer, steady-state processes are inherently easier to design and operate. And if you look at the chemical industry, the vast majority of accidents and incidents actually happen during the transient parts of the process and not during routine operations. If you think that through in our industry, where the way we operate our processes is still a very important factor in determining the product quality, having the ability to operate your process in a much more consistent way will actually enhance our ability to control our product quality better. So I would say that would be a compelling argument for our industry.
So what is your, did I get it right? I mean, were you in small molecule synthesis at first? Or?
I have done continuous chromatography for small molecules for a good part of my professional career. Yes, that's true.
Right, so let's just, I mean, based on that, you're saying that there's a lot more we can learn on that, on the chemical side for that as well, yeah?
Yes, I think as we start to have a better ability to measure what we are doing and to have a better eye on our process and our product quality, we can gradually learn from what other industries have taught us in terms of process efficiency. And that's exactly what many of these continuous technologies are doing. They are inspired by what people in dairy industries and chemical industries and food industries are doing already for a couple of decades. And we transform that into a technology that is suitable for our industry with its unique features, including the need for aseptic operations and all that. So that's what we try to do. We try to translate, I would say, commonly accepted operations in those industries and make them suitable for our industry.
This Lynn and Mark together. I mean, one of the biggest problems we've had is, so what is the process signature? I know on our end of the business, it's like we either lack the ability to measure it, don't understand what we're measuring, worse yet. And you know what I'm saying? In other words, what's a process signature look like? What's my end game? What's my CQA? And usually people start to like, oh my God, you know, that's the same reason QBD doesn't seem to be like the banner. Bottom line is, I guess we're a little off the main topic, but the bottom line is there seems to be skill sets, there seems to be knowledge and that have to be added into the process mix, that have to be provided. Most innovator companies realize that maybe they do or don't want to make that investment. So from a contracting or a supplier standpoint, what part of that do we get? If, you know, how do you fulfill that? How do you fill that gap? We'll start with you, Lynn.
Sure, yeah. Well, we work with companies on programs that look at fully continuous processes, and we try to help them through the next set of hurdles. So for instance, you know, there's really two main trends right now in the industry of how continuous is being adopted. There's a set of companies that are going full bore into fully continuous, everything from perfusion all the way to drug substance, and trying to link the entire process and run it for 30 days. And of course, that's a challenge due to aseptic operation and keeping things clean. And they're doing it in process development. It hasn't been done in manufacturing yet as a full-on continuous process. So this is a fairly new idea. And then there's the other group that are kind of looking at still traditional fed batch bioreactors, but using continuous downstream processing, or maybe just taking a segment of the downstream process, say from clarification. We have a new technology that's acoustic wave clarification that really reduces depth filtration, then sterile filter, then onto an inline concentration step, directly onto a multi-column chromatography capture step, and even semi-continuous viral inactivation. So if you look at that part of the process, so it's kind of pre-polishing in an average MAB platform, running that part continuous might have enough of an economic benefit or take care of a bottleneck in an existing facility that it's good just to employ that part of it. So some customers choose to adopt things in a very incremental way. It might only be 1 unit operation. Maybe they just want to get rid of their 1 meter protein A column and run a series of small 20 centimeter columns, because then they can cycle these columns to their lifetime within that one clinical campaign, and they can throw it out so people aren't storing protein A during clinical. So we'll get into the economics of that a little later, but I think those are some of the points of how things are being adopted.
So what about again, just what I'm saying is, it seems to me that the institutional knowledge folks like yourself have can be leveraged to our advantage significantly. I mean, you see what I mean? You get, right? So my point being is, is this something that process selection, continuous versus batch, this is something that you have the conversation with on a regular basis? That's where I'm at.
Yeah.
That's correct, yes. Yeah.
Yeah.
So many of the companies we talk to and who are inspired by this idea of continuous chromatography or continuous manufacturing as a whole, they normally know how to operate batch processes, and they are looking for a way to make those processes more efficient. So they typically are looking for a platform process that can accommodate multiple monoclonal antibodies or recombinant proteins, just like they have developed their batch process as a platform process, and they've characterized their process to the extent that they have built-in assurance that product quality is secured within the sort of design space or design window in which they operate that process. The conversion of that design space or design window into a continuous process is actually not something that is very hard to do once you've figured out how the translation of a batch process into a continuous process actually works. So the design space of a continuous process is not necessarily so much harder to design than for a batch process, but it requires the engineering mindset, which many of our customers are having right now.
So so where this is at, I mean, for example, so you're talking, that's where the single use throughout the whole process, that concept?
That eventually is I would say, the path forward. And that means that the conversion of a batch process to a continuous process will include a redesign of some of the skids that are required to run those processes, and they're going to be smaller, so that actually enables the design of these kits into a single-use format, which is appreciated by many of the customers. It's been one of the big revolutions over the past 15 years. So if you have these more intensified processes, then the whole single-use assembly becomes a reality for, I would still call it large-scale manufacturing, large-scale in terms of output, not so much large-scale in terms of the process equipment. And if you think about what Lynn just referred to, that very large-scale chromatography column, it now is reduced to a couple of very small columns. That saves about 75% of that very expensive chromatography media. So rather than having $5 or $7 million of chromatography material sitting in a column to accommodate a batch process, that can now go down to 20 to 25% of it in a continuous process, and the whole disposable design becomes a economically viable option. So the whole key of what continuous bioprocessing brings to the biopharmaceutical manufacturing is a significant reduction in the process equipment volume, and that enables the single-use design.
I got you. So Lynn, pretty much your standardization is what you're going after, right?
Yep. Well, and the other thing that we like to look at is, people often ask me, where's the cost savings in all this? Because there's a lot of things I have to change, in my plant, in my equipment, in people's training. And there are two kind of big cost savings in the standard MAB platform that we see. One is what Mark just mentioned, which is the chromatographic media cost. That's number one. If you're running protein A, that'll be the biggest cost saver. But surprisingly, when we've worked through some of these economic models with customers, the next big cost savings was actually quality, to the quality department in a clinical operation. And this is obviously forward-looking because this doesn't exist yet, but people who are looking to real-time release or at-line release, getting biomolecules to the point of where small molecules kind of just got to, and trying to be able to release the product when it gets to the end of the process. When we model that, there's a savings in QC cost that's probably the next biggest savings. And so that's the real big picture. It's hard to take a standard economic model and capture all of that today in a lot of models. And so I think people miss the point when they say, I'm only saving in chromatographic media. It's an operational savings that's not always easy to quantitate.
All right,
So-
a couple of things I heard there, maybe we want to go back. So when you go, and some, like, let's talk about the purification steps again. I mean, so like, the purification steps and the system design, can we touch on that, and how does that play when you go to continuous flow, for example? Did I ask that? Did I screw you up or what? I mean,
No,
okay,
not at all.
He invented it, so you know, for single
I'm
use.
looking at him like, whoa, what's happening here?
Well, I'm figuring out how to answer you in the limited time space we have. Well, what has happened is, for instance, for the chromatography system, if you want to overload the first column and capture everything that breaks through in the second column, which is something we do in continuous manufacturing, meanwhile other columns being loaded and being washed and diluted and equilibrated, that requires flow paths which are inherently different than which is common in batch chromatography systems. So inherently the whole valving system and the whole delivery of the fluid, the different buffers in the system is going to be different than what you expect in a batch chromatography system. So what our company has designed is a cassette that holds all the valves in a single-use device to accommodate all the controls of the fluid flows through the chromatography system. And that's a unique system that helps us designing a system that looks relatively complex, but inherently by its design becomes very natural and very well organized with a high segregation of fluids. And it's in a disposable format, so it's currently in the INTERPHEX in our booth. How this system works, it's by nature, these flows are completely segregated. Dead volumes in the cassette have been eliminated, and then it improves scalability, reduces complexity, and provides, I would say, predictability across all scales of the operation. So we've demonstrated from very small scale to relatively large scale that predictability of the performance is secured.
So, the reason I bring it up is Manny was talking about bottlenecks before, and just happened to touch on protein. So, do you want to? I think can we drill into like how does this fix this bottleneck?
Well, if you look what has happened in the industry over the past, say, 15 or 20 years, the science has progressed tremendously. So 20 years ago, you needed about a 20,000 liter bioreactor to produce commercially relevant amounts of products. Since then, the upstream process has been progressed so dramatically that nowadays we are at, say, 3 grams per liter, whereas originally it was only 0.2 grams per liter. So in the tank, we have at least 10 times more protein. And as a result of that, the downstream processing capacity has become the bottleneck rather than the upstream capacity. So originally we had all these reports out there that expressed the capacity of our industry in terms of the amount of bioreactor volume installed. Now the bottleneck is actually no longer the upstream capacity, but it has shifted towards the downstream. And that's where continuous manufacturing can solve the bottleneck, because you can't simply build a column wider than 2 meters. And that's what would be needed in order to address that bottleneck. So rather than trying to scale up or beat the physical limitation that we've hit with the downstream process equipment, going to continuous actually circumvents those limitations, and all of a sudden you can do the same job which you were originally doing with a 2 meter column. You had to cycle that multiple times to cope with all the product amounts. Now you can do it with 30 centimeter or 20 centimeter columns, which is I would say, but much better manageable product column size. So that's what continuous bioprocessing can actually do. What it also does, it eliminates the interstage tanks from between different steps. So if you build an island of continuity, the interstage tanks disappear, and those don't add any value to the process at all. So that's another bottleneck that's being addressed by building an island of continuity. The last thing that you could consider in the view of continuous bioprocessing is the fact that all these systems have multiple sensors which are detecting these repetitive patterns, and continuous operation has repetitive patterns or steady state patterns. all that data, if you turn it into information, you have a much better ability to control your process. And eventually, if you could correlate that to product quality attributes, real-time release and real-time testing becomes a viable option. So those are, I would say, the bottlenecks that continuous bioprocessing could solve.
There was a couple of comments from the prior speakers about the analytical capability for this. I mean, it continues to be a problem area. Well, you see what I mean for inline process control.
Yes, it is absolutely true that having inline and outline sensors is a challenge, although there's a lot of progress in that field as well, including spectroscopic measures, etc. What we should not overlook is the strength of all the information we already have. If you think about a chromatography process in a batch-wise mode, we normally have 1 or 2 elution peaks. We compare that to each other and say, well, our process has been operating consistently. Well, n equals 2 for statisticians is not a big deal. Right now in continuous bioprocessing, we may have, say, 40 or 50 elution peaks which can be compared to each other. So we have a much better handle on process consistency without having to add new sensor technologies. And obviously, if new sensor technologies become available, that will accelerate the ability to go towards real-time release and real-time testing, real-time release. But even with the information that is available today, we already can characterize our process and with that our product quality to a pretty reasonable extent, provided that we put the right, I would say, data analysis tools in place. And that is something that is much more relevant for a continuous process than it is for a batch process, simply because the amount of information that is made available is so much higher.
And by that, you mean the analysis of the assignment of a process signature, for example?
That is correct.
I mean, that's a never-ending debate. So Lynn, how's the pickup on this whole continuous process thing going here? I mean, we could talk till we're blue in the face. The bottom line is, where's the rubber hitting the road here?
Yeah, I think large companies have made some pretty large investments in prototyping what this can look like in a full manufacturing environment. And there's 2 kind of keys that are still being worked on aggressively, and that's the sampling. So you might have surge bags in between process steps in order to account for process upsets. You have to have an alarming capability where if something goes wrong in this step, you need to alarm back through the system because everything's not occurring in a batch and collecting, right? So these are things you have to think about. The other thing is automation. You have to have automations and controls that perhaps in the next generation can adapt to the incoming feed. That's kind of the holy grail of continuous manufacturing. If you really want to run continuously in control, you have kind of an operating parameter window space, and you have the actual line make the adjustments as it goes. So if you can get to the point where there's technologies available that feed back and allow, say, your chromatography skid to select a low, a medium, or a high titer method based on what's coming out of your perfusion bioreactor, then you've achieved something actually more valuable than what we do in batch today, kind of a smarter process, right? So these are things that are already presently prototyped within large pharma companies. A lot of people think this is a long ways away, but a lot of people are working on it very diligently. There's been public presentations by Merck and Company out of Kenilworth, by Bayer in Leverkusen, Germany, and some of these strategies also hooking to central DCS systems, figuring out how this control is going to work. So they're pretty far along.
So, yeah, there was a couple of guys with upstream, down. Where are we going with the hybridization of process? I mean, what's our take on this one? I mean,
I think, again, there's these 2 classes of people. There are the ones who are kind of from the ground up, from day one, saying we are building a fully continuous process from perfusion, running 30 days. This is the model. And it's the model either because the molecule needs fast processing because it's a labile molecule, it's an enzyme, there's some other reason, or because they've done the economic model and figured that running at this kind of 2 grams per hour, whatever the line will produce, will give them manufacturing flexibility that's needed for that product, right? That's kind of one school of thought. The other is, how do we just debottleneck our present process? And that tends to be the people who have fed batch upstream and need an answer for downstream. So if you think about downstream, if you're in a clinical facility running, and this is typical, I think, 10 to 12 monoclonals, let's say it's a monoclonal place, just as ease of example. If you look at what the Protein A spend is in a facility like that, just on that one consumable, it can be $10 million a year, right? Because they're often not running it again. So if you take that number and you can decrease that just in that one unit operation, again, you can see savings. So again, it depends on what the economic drivers, like Parrish was saying earlier, the business drivers for that particular facility.
So where do you see this going? I mean, what's the future here? I mean, I'm hearing big cost savings, I'm hearing steady state, but I'm also hearing I'm not really, you got to understand, I just came off the other panel, This is kind of what we talked about a little bit, So where are we going? What's your take on this? I mean,
Well, the earlier talk was originally targeted the future of biosimilars. We do see more and more products being approved in the regulated countries in Europe and US, and among them there are actually some biosimilars. There are also biotech drugs which target the same indication. So that tells us that we will see more and more products which require smaller annual demands, which have smaller annual capacities. So therefore you could expect that actually the capacities or the batches are getting smaller in the next couple of years rather than larger. So the whole promise of large-scale manufacturing may not necessarily mean that we are having a single product facility producing a ton of products of a single kind. We'll see much more facilities that are targeted to produce in a very efficient way multiple products in a very consistent way, and that's where continuous bioprocessing and single-use bioprocessing actually can work together to provide that flexibility and efficiency that is needed to address the need of those different products. So scalability and flexibility, scalability either by simply exchanging the single-use components, thereby addressing a different scale of operation, or by making the time a variable. Those are flexibilities that continuous bioprocessing has to offer, which could actually add value to the operational flexibility of the entire manufacturing facility. So the key to the equation remains making the process efficient and compact, and thereby promoting or enabling the single-use design of the entire manufacturing plant without sacrificing product quality or process efficiency.
I mean, it seems to me, just having spoken about it a couple of times, it's anybody who scales up and they use the conventional approach winds up with a building full of boat anchors eventually. I mean, because they lack flexibility. I mean, the way I look at it is if tomorrow I want to make B instead of A, I'm screwed. The bottom line is it would seem to me single-use flexibility, take continuous out of the equation, right? I mean, let's talk about it. I mean, isn't it more flexibility?
Well, even with the diversity of product pipeline, I mean, that's really driving the flexibility. So the days of just the one monoclonal, blockbuster, there are a few coming down the pike, but there's going to be more bispecifics, more kind of fusion proteins, lots of different recombinant proteins, more vaccines that are a little bit different technology. We're working with somebody on continuous on an algae cell culture for vaccine for flu, so there's all kinds of things going on in development that I think offer a very wide diversity, which is going to mean that you need the most flexible kind of platforms that could not only do protein A, but do ion exchange chromatography, can run membranes in a multi-membrane mode, can look at some interesting, even size exclusion can be done efficiently with multi-column chromatography. So you have to look at it as an opening up of perhaps the applications that will be needed in purification. We've kind of lived through the age of platform processes, and maybe now it's going the other way. We need more flexibility in our processes.
It would seem to me, I mean, especially when you consider where CMOs are weighed in. I mean, now, and they're they should own flexibility. I mean, because tomorrow, who's going to walk through the door?
Yeah.
I mean, where do you see, I mean, bottom line is, so you see this not only on the innovator side, you see this on a contract side. So what's the market look like? Who would like
so
to
right
address
now,
that?
I mean, right now the CMOs are looking at the technology. They obviously are reluctant to adopt anything until they have a paying customer because that's the business model.
Yeah, right.
But what I've seen personally, because I've been in this now a number of years, is that there are many more CMOs looking at this because the customers have come to them and asked them, do you have a way to evaluate continuous chromatography in my process? Or do you have a manufacturing answer if I were to develop a continuous chromatography step? So we are seeing more pull in that area right now. Yeah.
Is that pretty? I mean, there's at least 2 or 3 on the small molecule side. These guys are set up and ready to go now.
There will be a few announced this year, and one perhaps as early as June that will do a public announcement about an effort to offer continuous bioprocessing as a service. Yeah.
I got you. So how far downstream is continuous? Right down to fill finish? I mean, is that a fair question? I mean, obviously we don't sell it in 500-liter tanks, right?
No, I think it is fair to say that many companies are looking at continuous downstream processing until drug substance. So it is not uncommon to also hand it over then to a different facility. So then to sort of take a break after a drug substance being formulated, that is a fair point to say that's where the end of that continuous downstream processing today will end. I'm not saying that will be the case tomorrow, but today that's the primary focus of what we as a platform would like to offer. And it's going to take us quite a bit of effort to already take it up to there.
Right. I mean, it just seems to me that seems to be something we always play fast and loose with on the bio side. You know, to us, we haven't got a product like got a dosage form.
Right.
So we're pretty, if we were on my end of the world, we're talking about chemical synthesis here.
Yeah.
Okay, so where's the dosage form come in?
Right.
So what I'm saying is there's got to be somebody look, because that's got to be labor intensive, that's got to be high risk, we're talking a lot. So,
But traditionally, that's been a separate facility for a lot of folks.
and again, I'm not asking you to go off the
yeah,
out of it, but what I'm
yeah,
saying,
yeah.
there's got to be, as you know, with suppliers like you said, I mean, this has got to be something somebody's going to start to address. Because intertank, interchange, every time you move something, you run the risk.
Yep.
Right?
Yep.
Where does stuff go bad? Stuff goes bad with the infamous in-process storage, right down the sewer, right? I mean, that's usually where that winds up. Or, God forbid, when I'm shipping in bulk, I mean, that's the key, right? So,
Yeah.
where do you see, and again, I've asked every speaker this, where do you see this going? Hey, let's go fast forward here about 10 years. Where do you see this?
I think there's going to be a miniaturization of the GMP environment where we're almost into tabletop manufacturing, and that's a term that a couple of companies have thrown around over the years, where we're used to traditional GMP skids on wheels in a clean room. I think maybe 10 years out, you might see what looks like process development equipment, but obviously manufactured a little bit differently for product contact and having the correct GAMP compliant software to run in a tabletop environment. And if you look at the lab we're running for continuous in our PD area, we are now running in a PD area 2 grams an hour, even up to 8 or 10 grams an hour. On a PD scale that's set up in a big U, it's just PD equipment now. But if you took that same scale of equipment and made it GMP ready or a little bit different design, et cetera, set up for operators a little bit differently, you can envision just a room where the whole downstream process takes place in a 6 meter by 6 meter room. So a very different environment. That's where I think that I'm an optimist.
I'm there.
Looking
No,
ahead,
but
that's where I think it's going.
To me, the answer is if you do things the right way, in principle, where you make it should not matter because it's inside of a box. And this should be one. And this is something that the guys in drug shortage are dealing with. In other words, you put it on. If you could roll it onto an air freighter, you're in business. So. But to do that, that's where the integration comes in. Because quite honestly, one of the other things we're hearing in personalized medicine, it's going to fall. My opinion, it's going to fall to biotech. All of a sudden I come up with this new form, and tomorrow we make this discovery. This could be customized, patient-centric medicine, where the small is going to be the answer, right? So, how small is small? Let me ask you that. We've asked how big is big, but we're small. I mean,
That's a very good question. One of the challenges with continuous bioprocessing is actually to scale it down to the level where you can still manufacture reliably and not have the burden of the dead volumes be too much of a waste. So how small is small? I don't think with all the microfluidics that are currently being developed in the industry, it's very easy to answer, but I think we're gonna see further miniaturization of this of this whole continuous downstream processing. I'm not saying we're going into the robots area, but it is going to be smaller and more portable, so that indeed localized, standardized manufacturing is going to be an opportunity.
yeah, I mean, and I gotta say, the reason I picked, there was it's gotta be back about 2 months ago, there was an article where the guy was doing at-home biosynthesis, and I had to open it. I mean, what is this doing here? And it was like reading an excerpt from either Home Brewing or Breaking Bad. I mean, what is this guy? Turns out, and it was a person, a clinician that was interested in addressing the need for a very specific molecule, and said that there should be regulations and or equipment capable. And I'm thinking, well, that's an extreme extension of personalized medicine. But that was where the genesis, because it made me stop and wonder, so how small is small? And, right? How far do we go, So
I think there will be the one patient, some of these T-cell therapies and other things that are coming out for rare cancers, and there's going to have to be individual manufacturing that's been tried. There was a Boston-based a company years ago that was manufacturing for each patient in the hood. That wasn't very efficient, but of course closed systems weren't available then, so single use has leapt forward quite a bit since then.
Yeah.
So I think there will be kind of gene therapy is another example, but there will be many of these ideas that come around again. Sometimes it takes us twice in this industry. Ideas come in and then they die, and then they come back
I think in a lot
the
of
second
cases,
time and make it.
or they don't look around. Nuclear
Yeah.
pharmacy's been around for, right, and that's
Yeah.
still something that's made per patient.
Yeah.
I mean, not that I want to get into it, no, but anyway. But anyway, we're just about out of time here, so I'm sorry, I got to the point where I forgot everybody else was here. So are there any questions that we can take? We got a couple, don't everybody jump up at once like I often say. Okay, I'll take that as a unilateral, great job done, thank you.
Thanks.
Well done.
Pharmaceutical Processing, in conjunction with INTERPHEX 2016, presents INTERPHEX Live.